Hello everyone, thank you for having me present my project. I'm Mariem Ghoula, currently pursuing a PhD in bioinformatics at the University of Paris. Today I'll be presenting our project group work, Identification of Druggable Pockets and Structural Analysis of the Interaction between the SARS-CoV-2 Spike protein and the human ACE2 receptor. The spike protein or glycoprotein is the key element that enables the viral entry. Its structure is shaped as a homotrimeric protein, mediates attachment to the membrane of the host cell, and can trigger fusion between the envelope and the host membrane. On the other hand, the ACE2 receptor is known to be the main entry point into cells for some coronaviruses now. So as you might know, the spike protein is a trimeric protein, and the key element in its structure is the receptor binding domain, or commonly called RBD. And this domain needs to bind ACE2 in order to trigger the virus entry. And what's interesting is that the spike protein can switch from an inactive lock state to an active one. So essentially, when spike is inactive, RBD is unreachable, and that's what we call the down state. However, when spike is active or is in its up state, RBD is shaped in a way that ACE2 is not out of reach anymore, and that's what we are focusing on. So the main goals of this project is to identify key amino acids or hotspot involved in the RBD-ACE2 interaction, but also druggable pockets on the RBD domain to guide us in the discovery of inhibitors directed against the spike protein. So for this project and for feasibility, we decided to focus on the wild-type RBD and ACE2 complex, as it's a smaller system and can be easily studied. So we started with molecular dynamics simulations of both PDB structures. An MD is a computer simulation method for analyzing the physical movements of atoms, and it gives us a view of the dynamics evolution of a system during a fixed period of time. So it's more like a realistic representation of how the protein acts in physiological conditions. And in our case, MD was useful to examine the dynamics of the complex and the RBD in order to investigate their flexibility and also detect any structural changes that can be observed during the simulation. But MD was mostly important for both MMPBCA and POC drug methods that I'll be introducing later. The MD method was applied on a total of 4 systems, 2 systems including the RBD-ACE2 complex from both PDB structures, and 2 other systems of the RBD alone that has been isolated beforehand from both PDB structures as well. So 20 replicas were run for each system, which ended up with 8 microseconds total. MMPBCA stands for Molecular Mechanics Poisson-Boltzmann Surface Area, and this method calculates the free energies of protein-protein or protein-ligand system generated with MD. And what's interesting with MMPBCA is that it's also a tool that calculates the per residue decomposition energies to provide information about the local interactions of a system. And in our case, it was helpful to identify the most contributing or not residues on the RBD binding surface that we also call RBM for receptor binding motif. And finally, we use POC drug to detect druggable pockets on the RBD domain. So a set of 1,000 frames were selected from the MD simulations, and in total, we extracted 4,000 pockets. And the criteria were that only pockets comprising more than 10 residues and at least 1 hotspot are analyzed. Why more than 10? It's because a protein pocket is considered druggable if 10 or more amino acids are assembling it. And of course, they were selected based on their druggability scores and also classified, and we made sure that we analyzed stable and frequent ones through the MD simulations. With that, we identified 9 residues contributing with an energy equal or below -1 kilocalorie per mole. And then each of these hotspots were analyzed to see in which type of interaction they were involved in. So typically we had to see if it was hydrogen bonding, salt bridges, and each time we had to look with which ACE2 receptor residues they were interacting with. However, one of the residues on the RBM, glutamic acid 484, stood out during the MMPBCA analysis. And as we can see here, its decomposition energy was reaching highly positive values, which means that this amino acid is totally unfavorable to the complex interaction and therefore identifies as a negative hotspot. Which is also interesting is that the specific residue is found in 3 different COVID mutations belonging to the South African, Brazilian, and Japanese variants and exists under a complete different amino acid type, which is a lysine, this time acting as a positive hotspot. And our hypothesis is that the complete switch from a negative amino acid to a positively charged one, a lysine, might be the reason why these variants witness an escape from antibodies and develop a viral adaptation to the immune system. So finally, we ended up with 9 protein pocket classes. The druggable ones are shown below in a pink color. The more intense the color, the more druggable the pockets are. The second legend means the pockets are on the RBM if it's mentioned, otherwise they are located elsewhere on the RBM. So 4 classes of protein pockets are on the RBM, and 5 of them are located on other regions of the protein. We have an example of a pocket of the 8th class, which is located on the RBM this time, which is also druggable and comprises 3 hotspot residues. And for instance, it can be very interesting to use this pocket for further docking studies. And here we have another example of a pocket. This pocket belongs to the 5th class, and it's not located on the RBM this time. This pocket is 100% druggable since it's assembled with hydrophobic residues and is located pretty deep in the protein. And speaking of which, this pocket coincides with a binding region recently studied by researchers at the University of Bristol, and it comprises 5 common residues with the pocket we found. And this pocket is known to bind linoleic acid, which is a free fatty acid that unlocks the spike protein in its down state or inactive state. So it's another interesting target for future analysis and can be another strategy to inhibit the spike protein. So as a conclusion, 9 hotspot residues were identified as most contributing elements of the RBD, which then were spotted in interesting druggable pockets that can be analyzed in depth with future docking and screening methods. And as for our ongoing projects, we are currently working on the other COVID variants to see if there are any differences regarding the free energies analysis with this wild-type version of the RBD. And it's also important to see if other possible hotspots might arise in the presence of these mutations or even predict future ones, which can be very helpful in order to understand the complex mechanisms even better. And in addition to that, we will also be testing some inhibitors on these selected pockets and see if we can find a few interesting candidates to inhibit the spike protein, and also study the interaction between the RBD and some antibodies. Thank you for your attention, and thank you for having me again.